Hello everyone and welcome to our module on gluconeogenesis. On the right side of the screen here, I've shown the reactions by which cells in the body convert glucose into pyruvate and in the process of doing so generate ATP for energy. And gluconeogenesis is the process of going in reverse. So it takes molecules of pyruvate and converts them back into glucose. This costs the cell energy. The cell has to use ATP in order to go backwards, but this is a way the liver can generate glucose, which can be secreted into the blood to support other organs during times of starvation. And it's also a way that many other types of molecules can be converted into glucose for energy. So for example, other sources of carbon can be used by gluconeogenesis to make glucose. Lactate, some amino acids, propionate, which comes from odd chain fatty acids, and glycerol, which also comes from triglycerides. All of these can be converted into pyruvate or other intermediates of gluconeogenesis and then converted back into glucose so that cells can use the glucose for energy. So let's begin by talking about pyruvate. It's a very important molecule and it is the end product of glycolysis, and there are a number of different biochemical pathways by which the body can metabolize pyruvate. In skeletal muscle, pyruvate can be converted into alanine, and then there is a cycle called the alanine cycle where the alanine will be transferred back to the liver and the liver can convert it into glucose. We'll talk more about this in the module on amino acids. Pyruvate can also be converted into lactate by anaerobic metabolism, and lactate from skeletal muscles and red cells can be secreted into the plasma and then it can go back to the liver where it's converted to glucose. This is called the Cori cycle. The highest energy yield in terms of ATP from pyruvate comes by converting it to acetyl-CoA and sending acetyl-CoA into the tricarboxylic acid cycle, and we'll talk about that in a separate module. But another potential fate for pyruvate is to use it to go backwards via gluconeogenesis. So let's zoom in on the two major metabolic pathways of pyruvate, and that involves either going into the TCA cycle or going into gluconeogenesis. If pyruvate is metabolized via the TCA cycle, that will generate ATP. On the other hand, if pyruvate enters the gluconeogenesis pathway, that will cost the cell ATP because it requires energy in order to convert pyruvate back into glucose. So you can imagine that it would make no sense for a cell to be simultaneously metabolizing pyruvate by both of these pathways. It would be generating ATP by this pathway over here and then spending it on gluconeogenesis, and it would be wasting its efforts. So the cell wants to go one direction or the other. So how does it do that? Well, it does that by using acetyl-CoA as an indicator of the metabolic activity of the cell. The TCA cycle generates a lot of ATP. That's why the ATP level goes up when pyruvate is metabolized this way. And ATP is an inhibitor of many of the enzymes of the TCA cycle. That means that when ATP levels get high, the TCA cycle becomes inhibited and levels of acetyl-CoA begin to rise inside of a cell. Well, it turns out that acetyl-CoA is a very important activator of pyruvate carboxylase. It's called an allosteric activator because it changes the conformation of the enzyme and makes it more active. This is a brilliant system. It means that when there's a lot of ATP around, the TCA cycle will be shut down, acetyl-CoA levels will rise, pyruvate carboxylase will be turned on, and we will start metabolizing the pyruvate into gluconeogenesis so it can go back into glucose and supply other cells of the body with energy. So a key point here is that pyruvate carboxylase is inactive without acetyl-CoA. Acetyl-CoA is such a powerful allosteric activator that the enzyme has no activity unless acetyl-CoA is around, and that is how the cell makes sure that it's not simultaneously doing both of these pathways at the same time. So once a cell has decided that it wants to commit to gluconeogenesis, the first step is to synthesize phosphoenolpyruvate. If we go back to our overview slide here, the last step of glycolysis is the conversion of phosphoenolpyruvate into pyruvate, and this is an irreversible step of glycolysis. So in order for gluconeogenesis to occur, the cell is going to have to use other pathways to get around this irreversible step and synthesize phosphoenolpyruvate from pyruvate. In order to convert pyruvate into phosphoenolpyruvate, cells use a two-step process. The first step of the process is catalyzed by an enzyme called pyruvate carboxylase. This enzyme adds a COOH group to pyruvate in order to create a molecule of oxaloacetate. This enzyme uses CO2 as the source of the extra carbon group that it is adding to pyruvate. It also uses ATP. And carboxylase enzymes that use CO2 and ATP always require biotin as a cofactor. We'll talk more about this vitamin in a minute. The second step of the process is catalyzed by an enzyme called phosphoenolpyruvate carboxykinase. It's often just called PEPCK. This enzyme uses GTP as a phosphate donor group to add a phosphate group to oxaloacetate and create a molecule of phosphoenolpyruvate. Now, most of gluconeogenesis occurs in the cytosol, but this first step, conversion of pyruvate to oxaloacetate, occurs in the mitochondria. The mitochondria then uses the malate shuttle to get the oxaloacetate out into the cytosol for the second step, and I talk in more detail about how the malate shuttle works in the module on oxidative phosphorylation. So let's talk in a little more detail about the vitamin biotin. Biotin is a cofactor for carboxylation enzymes, and these are enzymes that catalyze the addition of one carbon groups via carbon dioxide. Specifically, they catalyze the addition of a carboxylic acid group. A carboxylic acid group is a COOH, and this is what a carboxylation enzyme will add to a molecule in the reaction that it catalyzes. There are three important metabolic enzymes where biotin is a cofactor. The first one is pyruvate carboxylase, which we're talking about in this module, also acetyl-CoA carboxylase and propionyl-CoA carboxylase. If we go back 2 slides, we can see that pyruvate carboxylase adds a carboxylic acid group to pyruvate, and it adds the carbon from CO2, and it uses ATP. And anytime you see a carboxylase enzyme that uses CO2 and ATP, it is going to require biotin for a cofactor. All of the carboxylase enzymes that require biotin as a cofactor use these two molecules in the reaction that they catalyze. Deficiency of biotin is very rare because the vitamin is widely distributed in the foods that we consume. One way people can rarely get a biotin deficiency is from massive consumption of raw egg whites. You need to eat an enormous number of raw egg whites in order to become biotin deficient, but raw egg whites contain a glycoprotein called avidin, which can bind to biotin and make you deficient in the vitamin. The symptoms of biotin deficiency are dermatitis, which will be inflammation of the skin, glossitis, inflammation of the tongue, loss of appetite, and also nausea. Very rarely children are born with a deficiency of the enzyme pyruvate carboxylase, and they will present with failure to thrive, and they will have high levels of pyruvate and also high levels of lactate and a lactic acidosis. If we go back to our overview slide of pyruvate, when levels of ATP get high, the tricyclic acid cycle backs up, so pyruvate cannot be metabolized this way. The level of acetyl-CoA will rise, and that would normally drive pyruvate into gluconeogenesis, but because pyruvate carboxylase is deficient, this pathway is cut off. This means that pyruvate will be driven towards lactate, and therefore the child will have a lactic acidosis. They can also have a high alanine level as well, and that's because metabolism to alanine or lactate are the only two pathways open to pyruvate when pyruvate carboxylase is deficient. So back to our steps of gluconeogenesis. Once we have synthesized a molecule of phosphoenolpyruvate from pyruvate, we can then go backwards through glycolysis until we reach a molecule of fructose 1,6-bisphosphate. If we go back to our overview slide of glycolysis here, once gluconeogenesis has synthesized a molecule of phosphoenolpyruvate from pyruvate, it can then go backwards through the reversible reactions of glycolysis until it reaches the level of fructose 1,6-bisphosphate. This reaction right here is irreversible, so gluconeogenesis requires its own enzyme to get around this and synthesize a molecule of fructose 6-phosphate. Recall that the glycolysis enzyme that converts fructose 6-phosphate into 1,6-bisphosphate is called phosphofructokinase 1. The enzyme for the reverse process in gluconeogenesis is called fructose 1,6-bisphosphatase 1. And just like PFK1 was the rate-limiting step for glycolysis, fructose 1,6-bisphosphatase 1 is the rate-limiting step for gluconeogenesis. Because these two enzymes are the rate-limiting steps of glycolysis and gluconeogenesis, their relative activity determines whether molecules will flow in this direction or in the opposite direction. So when the level of ATP is high, the cell has plenty of energy and does not need to conduct glycolysis. Therefore, high levels of ATP tend to activate fructose 1,6-bisphosphatase 1, and they tend to inactivate phosphofructokinase. This will drive the flow of molecules in this direction. On the other hand, when AMP levels are high, this means that lots of ATP is getting broken down into AMP, and it means that the cell needs more energy. Therefore, when AMP levels are high, it tends to inhibit fructose 1,6-bisphosphatase 1, and it tends to activate phosphofructokinase. And this will drive molecules in this direction, and this will favor the breakdown of glucose into ATP to replenish the cell. And then a final molecule that has a similar function to AMP is called fructose 2,6-bisphosphate. This molecule drives things in the direction of glycolysis, and it tends to shut down the enzymes for gluconeogenesis. Fructose 2,6-bisphosphate is a very important molecule for the regulation of glycolysis or gluconeogenesis. As you may know, the hormones insulin and glucagon can determine whether cells will favor glycolysis or gluconeogenesis, and the way they manipulate cells is by manipulating the level of fructose 2,6-bisphosphate. The easiest way to keep this molecule's function straight is to think of fructose 2,6-bisphosphate as an on-off switch for glycolysis. When the level of this molecule is high, that favors glycolysis, it turns glycolysis on. And when the level of this molecule is low, that favors gluconeogenesis, it turns glycolysis off. The way fructose 2,6-bisphosphate does this is by manipulating the enzymes PFK1 and fructose 1,6-bisphosphatase 1. When it wants to favor glycolysis, it will activate this enzyme and inactivate this one. And when it wants to favor gluconeogenesis, it will do the reverse. What's confusing about fructose 2,6-bisphosphate is that it is synthesized as an offshoot of fructose 6-phosphate, one of the molecules whose levels it is designed to control. Just like there are a pair of enzymes called phosphofructokinase 1 and fructose 1,6-bisphosphatase 1 that are used in glycolysis and gluconeogenesis, there is another pair called phosphofructokinase 2 and fructose 1,6-bisphosphatase 2, which will synthesize fructose 2,6-bisphosphate so that this molecule can go and manipulate glycolysis and gluconeogenesis. All this can get very confusing, but if you just remember what I've written in this box on the bottom of the screen here, you can usually keep straight what's going on and what the cell is going to do in response to a change in the level of fructose 2,6-bisphosphate. In the glycolysis module, I talk about exactly how insulin and glucagon manipulate the levels of fructose 2,6-bisphosphate, but I'll just summarize that for our purposes here. So levels of this molecule rise with high insulin level in the fed state, and this tends to favor glycolysis. Levels fall with high glucagon in the fasting state, and this tends to favor gluconeogenesis. And as I said before, the levels of this molecule drive glycolysis versus gluconeogenesis. The step of glycolysis where fructose 6-phosphate is converted to fructose 1,6-bisphosphate by PFK1 is the rate-limiting step of glycolysis. The reverse step is the rate-limiting step of gluconeogenesis. So it's very high yield if you can remember what makes these two steps go or slow down. So I've drawn it on the screen as a stoplight with things that make each enzyme go and things that make each enzyme stop. So when phosphofructokinase 1 is activated, that will favor glycolysis, and things that activate this enzyme include the levels of AMP and the levels of fructose 2,6-bisphosphate. Things that slow this enzyme down include ATP and citrate. And all of this should make intuitive sense to you. The level of AMP will be high when the cell needs energy. The level of ATP will be high when the cell does not need energy. Citrate comes from the citric acid cycle, so when this level is high, it means that there are lots of mediators of the citric acid cycle, and therefore it will slow down glycolysis. The molecules that activate the enzyme fructose 1,6-bisphosphatase and therefore that activate gluconeogenesis are essentially the reverse of the other side. So when levels of AMP are high, the cell is wanting for energy and it does not want to perform gluconeogenesis and spend ATP to synthesize glucose. When levels of ATP are high, the cell has lots of energy and therefore it wants to do gluconeogenesis and synthesize glucose from other carbon sources. Back to our steps for gluconeogenesis, the final step in conversion of pyruvate to glucose is the conversion of glucose 6-phosphate into glucose. Back to our overview slide, once we have converted fructose 1,6-bisphosphate into fructose 6-phosphate, we can then go backwards through glycolysis until we reach glucose 6-phosphate. This is an irreversible step of glycolysis, so gluconeogenesis requires its own enzyme to synthesize a molecule of glucose. The enzyme that does that is called glucose 6-phosphatase, and it is found predominantly in the liver and the kidneys. These are the two organs that can synthesize glucose and secrete it into the plasma to supply other tissues like that of the brain. Other organs that do not have glucose 6-phosphatase can still perform gluconeogenesis. However, the process stops at a molecule of glucose 6-phosphate, which can then be shunted towards glycogen and stored for later use. Glucose 6-phosphatase removes this phosphate from the 6-carbon of glucose to synthesize a molecule of glucose. And importantly, this reaction occurs in the endoplasmic reticulum. They like you to know that for step 1 of your boards. So here's a summary slide of the steps of gluconeogenesis by which pyruvate is converted back into glucose. So for pyruvate to get to phosphoenolpyruvate, there is a 2-step process to get around this irreversible step of glycolysis. Remember that acetyl-CoA is a very important allosteric activator of pyruvate carboxylase for this step of gluconeogenesis. In going from fructose 1,6-bisphosphate to fructose 6-phosphate, there's a 1-step process. Remember that this is the rate-limiting step. It's activated by high levels of energy in the cell, represented by high levels of ATP, and it's inhibited by levels of AMP and fructose 2,6-bisphosphate, as I described before, and fructose 2,6-bisphosphate is where insulin and glucagon exert their effects on glycolysis versus gluconeogenesis. And then finally, the last step of gluconeogenesis is conversion of glucose 6-phosphate into glucose. Now, fructose 2,6-bisphosphate is not the only place where insulin and glucagon can exert their effects. There are other steps in this process where those two hormones have been shown to exert influence. Glucagon has been shown to inhibit the conversion of phosphoenolpyruvate to pyruvate. This slows down glycolysis. Insulin has the opposite effect. Glucagon has also been shown to activate the conversion of glucose 6-phosphate to glucose. Insulin has the opposite effect. A word about all the different substrates that can feed into gluconeogenesis in order to create molecules of glucose to use for fuel. So lactate and alanine can both be converted into pyruvate, and therefore they can enter gluconeogenesis and become glucose. Many amino acids, so-called glucogenic amino acids, can be converted to pyruvate or OAA in order to enter gluconeogenesis. Odd-chain fatty acids can be converted into a molecule of propionyl-CoA, which can then become OAA and enter gluconeogenesis. The words odd chain are very important here. Even-chain fatty acids cannot be turned into glucose. They can be used for energy via the pathway of beta oxidation, but they cannot be converted back into glucose. Only odd-chain fatty acids can become glucose. And then finally, the body stores fatty acids as a substance called triacylglycerol, and when it mobilizes those fatty acids, it releases a molecule of glycerol. Glycerol can be converted back into intermediates of the glycolysis and gluconeogenic pathway, and therefore it can be used to synthesize molecules of glucose via gluconeogenesis. Finally, I'll finish this module by mentioning some of the other hormones besides insulin and glucagon that can affect the process of gluconeogenesis. So epinephrine raises blood glucose levels as part of the fight or flight response, and it can activate gluconeogenesis as well as glycogen breakdown in order to do this. Cortisol can increase the gluconeogenesis enzyme levels inside of cells, and hyperglycemia is a common and well-described side effect of patients who take steroid drugs, which work in an analogous manner to cortisol. These can raise the blood sugar level, and patients who take long-term steroids sometimes can develop steroid-induced diabetes. And then finally, thyroid hormone also increases the activity of the gluconeogenic pathway and all its enzymes and intermediates. And that concludes our module on gluconeogenesis.